Greetings everyone. So today we are going to discuss about the endospore formation. First, we need to understand that uh, why are there certain bodies which are formed in unfavorable circumstances, and what do the bacteria do in order to survive unfavorable circumstances. So, in favorable circumstances, whenever there are certain given conditions, the bacteria usually have their normal metabolism going on. But in case of any sort of unfavorable circumstance, such as temperature change, high or low temperature, high pH, or chemical changes, desiccation, ultraviolet rays, or antibiotics being attacked on bacteria, they tend to change their structure and form metabolically metabolically dormant bodies. They form metabolically dormant and that's body metabolically dormant and resistant body. They form metabolically dormant and resistant bodies in which we have they will form metabolically dormant and resistant bodies. And these metabolically dormant or resistant bodies, which are formed during unfavorable circumstances of their growth, particularly during the late stage of growth, whenever there's depletion of nutrients during the late stage of their growth, they tend to form metabolically dormant resistant bodies which are known as spores. Now, spores, these metabolically dormant and resistant body involve examples of spores, among which we have exospore, endospore, exospore, endospore, as well as we have another example of metabolically dormant body which is known as cyst. We are going to first discuss about the endospore. As the name suggests, endospore, it signifies that spore which is produced within the bacterial cell wall, is known as an endospore, while a spore which is formed outside the bacterial cell wall as an outgrowth of the bacterial cell, is known as exospore. And there are certain species of bacteria which are associated with endospore formation. These species involve examples of clostridium, clostridium, and bacillus species. Clostridium and bacillus species form these are associated with endospore formation. And when it comes to the process in which the formation occurs, first and foremost, spore formation is a process which is known as spirulation, as we have previously studied. Sporulation, sorry, sporulation, and that sporulation process basically involves the formation of spores which are originated from a structure which is known as sporangium. 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 Right? So in this case, we are also having sporangium. Now, in this case, the bacterial cell which is producing the endospore would be considered as sporangium. So we can say that bacterial cell is also an sporangium. And after that, a cell which divides itself is known as a vegetative cell. So let's take a look onto how the vegetative cell forms endospore as a sporangium. Now, first step which is involved in the cycle is axial filament formation, such that there are some axial filaments formed. um, Axial filaments are formed by the invagination of the plasma membrane into the cytoplasm, which causes a formation of axial filament, right? So formation of this axial filament would be considered as formation formation of axial filaments over here. That here Axial filament formation. Axial filament formation due to the invagination of plasma membrane causes the cell membrane to divided to be divided into fragments, after which the second step is the formation of septum. The step cell is septated. uh, there one more thing, that before the cycle occurring of the endospore formation, the chromosome material is duplicated and separated, such that after the formation of chromosome duplication and separated, we will be having the axial filament formation, after which there would be septation of the cell occurring, due to which the cell will be divided into two structures. The smaller structure would be considered as the forespore, and the larger one, which is having the more nuclear material, would be considered as the sporangium. Now, um, third step basically involves the engulfment of spore, a forespore by the sporangium, such that the sporangium engulfs the forespore for further development, as a result of which it causes to produce an early spore, and that early spore basically now uh, begins to actively synthesize the spore layers around the forespore, due to which the the fifth step here is the development of the layers, and sixth step is the deposition of the cortex and outer coat, or the resistant thick protective layers around the spore, due to which these layers being deposited around the spore will cause to form an immature endospore. Immature endospore would be formed. Now, in order to maturate it, the immature endospore will have the lysis of rest of this cytoplasmic material, as well as the cell wall and cell membrane, due to which the rest of the structure which would be left behind would be the endospore, due to which the endospore now would be considered as a mature endospore after the lysis of the bacterial cytoplasm, as well as the bacterial cell wall and cell membrane. And when it comes to the number of layers, there are certain number of layers around the endospore, such that endospore has core, which is the innermost layer, thick resistant layer, after which there is cortex, after which there is outer coat. So this is how the layers are formed. And uh, this is about the formation of endospore. Moreover, endospore is highly resistant to temperature, desiccation, environmental harms. Desiccation, antibiotic resistant, as well as chemical resistant, drugs are resistant, heavy metals resistant. And when it comes to exospores are basically structures which are also formed during reproductive stage of the bacteria as well. Such that if we talk about, let's suppose there's a bacterial cell, this is a bacterial cell, and if there is any sort of unfavorable circumstances occurring during the reproductive stage, the bacterial cell will tend to grow an outgrowth. It will tend to grow an outgrowth outside the cell wall, just like the budding process occurring within yeast, which would be forming uh, an outgrowth, and that outgrowth would be considered as exospore. Now, that outgrowth exospore, due to the unfavorable upon the arrival of the unfavorable circumstances, will cause the rest of the lysis, will cause again the same process which occurred in endospore, such that the outgrowth which is formed outside the cell, the structure is a bit messy, but you can better understand it. So here the structure basically here is considered as exospore. I've just tried to draw the rough structure, and that exospore will now become matured upon the lysis of rest of the bacterial cell and cell wall, due to which that exospore will be formed. Now, exospore forming bacteria involves example of streptomycetes and actinomycetes. Apart from which, there is a special chemical present within these bacterial thick resistant spores, both endospores as well as exospores, which are known as calcium dipicolinic acid, dipicolinic acid, which causes to provide that sort of high amount of resistance against such environmental conditions. Calcium dipicolinic acid, also known as CADPA. CADPA, calcium dipicolinic acid. It is found both in endospore and exospore. However, endospore is more resistant Then exospore, reason behind it is that exospore is having a bit of its layer, thin as compared to endospore, such that if we talk about their layers, all of these layers are present. Both outer coat is present, cortex is present, as well as the outer retina is present. Outside the outer coat, we are having a sort of attachment of a layer which is known as retiny, something we call as retiny. I'm not sure about the actual spell, but retiny is a layer, and this layer basically is a bit thinner in exospore as compared to endospore, which is a bit thicker. After which, when it comes to cyst, cyst basically is a process, is also a metabolically dormant body, but in this case, the metabolically dormant and resistant body will be uh, formed within the cell, such that let's suppose if we have a bacterial cell, and this occurs in azotobacter species. Azotobacter species, cyst formation occurs in azotobacter species. Now cyst basically will uh, cyst basically cyst basically it is formed whenever there is the deposition of thick cell wall around the bacterial cell, due to which the bacterial cell is covered from outside with a thick protective coating, due to which it causes to form a structure which is known as cyst. And that process of formation of cyst is known as encystment. And when it comes to the resistance of cyst is less resistant as compared to endospore, exospores, and even though it can resist chemical changes, desiccation, but it cannot resist high temperature as well as ultraviolet rays. So in this case, if we talk about the resistance of all these dormant bodies, we can consider endospore as a highly resistant structure, after which we have exospore as the second highly resistant structure, after which we are having cyst as the lowest resistant structure. So these are some structures which are going to be discussed. Hopefully you enjoyed the video. We'll meet in another video.